Hi, I am Aislinn Borland and I'm based at the University of Strathclyde, and I'm going to talk today about some work we've done modelling the early marine migrations of Atlantic salmon, and then some future applications of this model to the small-scale movement of salmon around wind farms. So the overall motivation behind this is understanding some of the declines we've seen in salmon populations over the last 50 years. These are attributed to declines in marine survival rates, particularly during the first few months at sea. However, during this time, we have a real lack of knowledge on the migratory paths of salmon, and that makes it difficult to understand the specific conditions they experience. So, for example, if we can model the time spent in different oceanographic regions, we can evaluate what prey or predation conditions they might be experiencing. So salmon are known as post-smolts during the first year at sea, and post-smolts from around this region will migrate towards feeding grounds in the Norwegian Sea. So here we have modelled migrations from a group of rivers in Ireland, Northern Ireland and Scotland, orange dots on the map there. And modelling is over the first 3 months at sea. One thing that it's important to note that I'm going to come back to is that these orange dots are actually 30 kilometres away from the coastline. The reason being that we will actually need a slightly different model to simulate the initial small-scale migration. But for this part, we use a classical tracking model coupled with an active swimming model, where movement of fish is based on ocean currents, salinity, and directional or magnetic views. And this is forced by the SSWRS, 27-year reanalysis of the area in blue on the map. So the results of the model look something like this. This is the density of all of the modelled post-smolts from all rivers and all years. And I won't go into too much detail here. You can scan the QR code to go to our paper if you want to find out more. But we looked at inter-annual variation in migrations, finding that to be related to local surface currents and salinity. We've also found little difference in the outcomes of migrations between fish which left the rivers earlier and slightly later. But we did find differences in migrations between rivers, and some of those differences were related to the distance of each river to the continental shelf edge. So on the left here, this might be familiar. This is a map of planned and existing wind farms around Scotland. And something we might want to consider is whether post-smolt migratory paths overlap with these offshore energy structures. If so, we might want to think about how this influences the conditions they experience and if this relates to marine survival rates. So to start to answer these questions, we need a sort of fine-scale movement model. As I mentioned before, the type of thing we would need for that initial section of the marine migration. So it's kind of an adaptation of what we already have. The differences in the model here are that first we need a smaller time step. Previously it was a 12-hour time step, and we'll need something like an hour for this, potentially even less. The modelled fish will also need to use finer scale navigational cues and potentially, quite likely, different swimming behaviour of fish. So looking at this region shown in black here, some wind farms around the north coast of Scotland, the reason for focusing here was that telemetry work, interestingly, had revealed smolts spending up to 10 days in this region. So to see what modelling smolt migrations around here could look like, the very first step that we've taken is just simulating passive movement. So this is the fish just drifting with ocean currents. And this was using a 1-hour time step and just from 3 rivers in northwest Scotland, which were picked because we expect fish from these rivers to migrate through this region. So Again, this is a density plot of the modelled closed smolts, and you can see that there's a relatively high density within that box around the wind farms. And we also found residence times of up to about 10 days within that box, which is very similar to the telemetry results here, which is interesting. However, in reality, passive movement of smolts is quite unlikely. We know that passive movement cannot guide them towards the Norwegian Sea for their entire migration, so they still need a bit of work. The next steps here to improve our understanding of smolt movement around marine energy structures. Basically, we need to adapt the existing model. We had that initial look using passive movement, but now we need to consider what other simulated and more likely swimming behaviours of smolts can recreate the patterns that was seen, patterns of movement seen from the telemetry around the swim farm. This can also be compared to other telemetry data that we have. For example, there's been a lot of work around the west coast of Scotland tracking smolts. And a further application of this model will be looking at interactions between salmon smolts and aquaculture sites. So that's all from me. Thank you for listening.
Today I'm going to talk about the West Coast Tracking Project. So just to recap, in the life cycle of Atlantic salmon, adults are returned to freshwater to spawn. Their juvenile offspring then remain in freshwater for the first few years of their life before they undergo that transformation into smolts that are able to survive in seawater, where they migrate out to sea to reach those rich feeding grounds and feed and grow. And it's the smolt stage, that first migration to sea, that we were interested in for the West Coast Tracking Project. So based on previous work, we know that the salmon smolts are heading towards the Wiley-Thompson Ridge and the Voring Plateau Escarpment, and they're using that strong North Atlantic current to get there. But what we don't know, or we know very little about, is how salmon smolts emigrate from their river of origin towards the continental shelf or towards that strong North Atlantic current. And that's where the West Coast Tracking Project came in. We really wanted to understand the early phase of the marine migration of salmon smolts and use that information to inform the management and policy of the species. So it's a 3-year project starting in 2021 with the final data collection year being in 2023, and we're just in the process of analysing all the data collected and writing that up for scientific publication. The project's a partnership between Atlantic Salmon Trust, Fisheries Management Scotland, Marine Directorate, and has support from the University of Glasgow. And as I've said, the overall objective is to look at the timing, speed and direction of migration through sea lochs, nearshore coastal and marine waters. So to answer this, we're using acoustic telemetry. We're inserting a coded tag into the salmon smolts. That tag emits a unique coded ping that's detected by stationary receivers or listening stations that are deployed throughout the study area, and the receiver records that fish's unique ID as well as the time and the date, and we can use that data to start making inferences about the migration behaviours of salmon smolts. So I'm just going to go through the study design for the West Coast Tracking Project, changed over the 3 years of the study. In each of these maps, there's grey dots, which are the receivers deployed by the West Coast Tracking Project, and then the yellow dots, which are receivers deployed by other projects. So in 2021, we were focused on migration through the Minch and between the islands of the Hebrides, and we tagged 971 salmon smolts from 9 rivers across the west coast of Scotland, deploying 224 acoustic receivers. In 2022, we focused on our efforts around the island of Mull and the Inner Hebridean Islands. We tagged just over 1,000 salmon smolts across 9 rivers to look at this. And then in 2023, we focused all of our efforts in the sea lochs. So we'd been collecting data throughout the West Coast on movement through sea lochs, but we decided that we were going to put all of our efforts into looking at the migration behaviours of salmon smolts through sea lochs in 2023. So we deployed 374 receivers throughout the sea lochs that we included in the study, and we tagged 1,186 salmon smolts across 13 rivers to address that question. So I don't have time to go into many of the results, so I'm going to focus on one specific. I'm going to talk about the marine migration, and specifically in 2021. So here we've got a map that shows the migration pathways of salmon smolts detected from the West Coast Tracking Project on marine receivers, and each line is an individual salmon smolts pathway. Now we've got straight lines. Salmon smolts don't swim in straight lines, but it's just an illustration of where that salmon smolt was going from and going to and coming from. So we can see we tagged salmon smolts in the southwest of Scotland in the Bladnoch and the Nith, and they were detected migrating north through the Irish Sea. Salmon smolts from around the island of Mull in Loch Aber and Argyll were detected migrating both ways around the island of Mull, through the Sound of Mull and the Firth of Lorn. And once they left those waters, they were detected in multiple different places. They were detected, or a couple were detected in the Minch waters, migrating between Skye and Uist. A couple were detected migrating between the islands of the Hebrides. One smolt was detected on the continental shelf by a wave glider that was deployed by Sea Monitor. And one smolt decided to go southwest and was into the North Channel and was detected off the coast of Ireland. In the north of Scotland, salmon smolts from Laxford were detected on that northern array jutting off the west coast, and smolts from Laxey, which is on the east coast of Lewis, were detected on the array jutting off Lewis and the array jutting off the northwest. So they were detected on both arrays, so there seems to be that they're spreading across that northern part of the Minch. Now, I just want to put those findings into context of a paper that we published last year. Basically, I've shown earlier with those maps that there was multiple projects happening on the west coast of Scotland or the west of the British Isles at the same time. And we were able in 2021 to collate all that data together to produce this paper looking at the inshore and offshore marine migration pathways of salmon smolts from Scotland, England, Northern Ireland and Ireland. And we all had the projects all had different objectives, but we were using the same technology, so we're able to pull that data together. And between the 10 projects, we tagged 1,914 salmon smolts in 2021 from 25 rivers and deployed 414 acoustic receivers. So here's a map from that paper, and you can see where all those acoustic receivers were deployed and where all the fish were tagged, and I've listed here all the projects that were involved. Most were tagging salmon smolts, but there was a few like MES MPA and Samosas that were deploying receivers for other reasons, but because they're using the same technology, we were able to share detections. And that wave glider that was deployed by Sea Monitor off on the continental shelf that had an acoustic receiver attached, and it went backwards and forwards. So you can see that this really builds on what I've shown with the West Coast in 2021. We've got more pathways shown on this map. Really the key take-home messages from this was that there was no single migration route or pathway. Smolts were migrating in multiple directions. There was extensive between river variation as well as within river variation in those pathways. In 2021, no fish coming from the Irish Sea were detected in the Minch waters. Now that only applies to 2021. And our longest tracked fish was from the River Burshall on the west coast of Ireland, which migrated 564 kilometres in a straight line over 26 days. So thank you for listening. These are all the organisations that were involved with either the West Coast Tracking Project and that Pathways paper. It's really a huge collaborative effort, the West Coast Tracking Project, and it's thanks to everyone working together that we were able to find all these amazing findings. So thank you.
Hi, I'm Matt from the Marine Directorate, part of the Marine Renewables and Ecology team. I'm going to be giving you a brief presentation today on some acoustic telemetry work that we have been doing over the last couple of years. I'm hoping from the previous presentations you have an overview of what acoustic telemetry is, and I'm going to hopefully jump right into the data. The aim of this work was to look at the spatial and temporal distribution of salmon smolts, and that is a key evidence gap from the ScotMER evidence maps themselves, which is what is the spatial and temporal distribution of salmon from rivers in this region. And by region, we're specifically looking at the north coast. Although I'm here talking to you today about this work, it was put together as part of a team with Emily Bridcut, Robert Main, Jonathan Archer, and also Christina Murray. Some of the work that we're doing, the main work that we are doing, is focused on three quite basic principles. That is, where are the species? Do they interact with offshore development? And what is the result of that reaction? Through the work that I'm presenting today, we really are focusing on that first aspect of that part of the puzzle, and that is, where are the species themselves? First of all, I'm going to go back to 2023, and there's a much more detailed presentation on this data on YouTube at the ScotMER website, that goes into a lot more detail, but I'm going to go over it very briefly again because it's very useful for setting the context for what we did in 2024. So in 2023, we were able to expand on the West Coast Tracking Project, which is down here, which was run primarily by the Atlantic Salmon Trust and partners. They tagged over 1,000 smolts emigrating from many different West Coast rivers and some on the Outer Hebrides as well, with an aim to follow their migration going north. We saw an opportunity to deploy 21 acoustic receivers, which are these black dots here, across the kind of top of the Minch on the north coast of Scotland, that allowed us to build on that project and try and get some more information on fish further north than we've ever been able to follow them before, and been able to detect fish going into that part of the sea where we have virtually no information on what smolt behaviour is in that area. What we expected to see was that the fish would be seen very briefly, only for a few detections on their migration north, that it'd be quite an active migration, so we wouldn't see them for very long, and that they'd be mainly constrained to the west, which is the fairly direct route, or the most direct route for fish going north, coming out of this area and these rivers. We detected a total of 72 smolts across the array itself, 33 from the Laxford, some from the Snizort, Laxey, Grimmister, Loy and Lundy. And in general, that was probably far better than we anticipated. Each of these dots, or each of these receivers, is about 7 kilometres apart, and so it's what we call a leaky line. It'd be very easy for tagged fish to be able to pass through that array and not get detected. So detecting those fish was much better than we thought. And they were spread across the entirety of the array. You can see here where the yellow dots were all the fish were detected, and these black lines are just to give a bit of a picture of where the fish went. We don't know if this was the actual route they took. It was highly unlikely that it took a straight line to get to these areas, and these lines are not able to pass within 5 kilometres of the coast, just to give a bit of realism about the potential path that smolts took to get to this area from where they were tagged and released. And if we zoom in on a couple of the populations, on the left here we've got the Snizort, so fish coming from the north of Skye, and we can see that there's quite a big spread in where the detections were, and interestingly quite a few of those fish were detected right across to the east of the array. If we look at the Laxford, where we detected more fish, you can see that detected across the entirety of the array itself, but again there's a number of fish that were actually coming to the east of that array and not heading in what we'd expect to be the most direct route north to their feeding grounds. So next, if we look at the total time on the marine array, so this is the time from the first detection of the fish to the time of the last detection on the fish, we find some really interesting results. And what we see here from each of these graphs is that each of the black lines represents a day, and so what we'd expect to see is that the vast majority of the fish would be passing through on the first day of detection, that if that is an active migration. But what we actually see is across all of the populations, quite a number of fish is staying for much longer, or been detected for much longer, some of them up to 15 days. And this was about 25 to 30% of the fish that we detected were actually showing this behaviour, much longer detection on the array itself. And it was the same across populations. So as you can see there from the individual graphs, that there are fish from each of the different populations that are showing this behaviour have been detected for much longer on the array itself, which was not as expected. Fish should be heading north relatively fast. And when we moved on to 2024, we decided to deploy a grid array. This is at the same latitude as in 2023, so the middle line here is the same latitude of the line in 2023. That allows us to give as much repetition as possible between years while still being able to answer different aims. The reason for using it as a grid is to hopefully be able to pick up movements of fish going backwards and forwards within this array, rather than simply side to side or passing straight through it. We hope to pick up some individual behaviour itself. To note, there are a lot fewer fish tagged in this year. There were 65 fish tagged in the Laxford, of which we detected 29, and there are 116 fish tagged in Loch Torridon, of which we detected 17, which is probably as we'd expect, given that Loch Torridon is much further away than the fish from the Laxford would have to travel. So again, if we look at the duration on the array, and this is between the Laxford and the Loch Torridon fish. Remember, there's quite a fewer fish from Loch Torridon itself. Within the Laxford, we again see that there are quite a few fish that are staying for longer periods of time. Again, each of these grey lines represents a single day, and that there are a number of fish that are staying for quite a bit longer. However, we don't see this in the Torridon fish. The vast majority pass through within those first couple of days of being detected. There's potentially a difference here in behaviour, but remember also much fewer fish were detected from Loch Torridon, and maybe we just don't have the sample size to be able to pick up this behaviour that we've seen in Loch Laxford itself. When we look at the Laxford data between years, we can see that there's quite a similar pattern, that there are a number of fish that pass through quite quickly in both years, but again that there are also a number of fish that are spending quite a bit of time on the array itself, for up to 14 days in 2023, somewhere up here, slightly less but still extended up to 8 or 9 days in 2024. But again, there's quite a few number of fish that are staying within the area or being detected on the array for longer than we would have expected, potentially milling around, but certainly not a active directed migration within this area. And coming on to the inter-array behaviour, and this is what the grid allows us to do here. This is a busy slide, but what I want to focus on is the blue lines themselves. So if we were seeing a directed migration, we'd expect all these blue lines to be going in roughly the same direction, either heading north, northeast or northwest, but heading in roughly the same direction, that the fish would come in from the south, be detected somewhere along the grid here, and then move their way through, and final detections being on the northern part of the array there. But what you can see is that there are lines going in virtually every direction, some north, some south, fish going what appears to be round in circles. There is no consistent direction in the movement of fish here. They are going in many different ways, and what you can also see from quite a few fish is that it's not a consistent direction. So we've got this fish here that is heading north and then east and then southwest, and again similar for this fish, going almost round in a complete circle in the detections itself. But what we can also see is that because of the size of the grid, fish are obviously able to get through that grid without being detected, is that although we are picking up quite a lot of behavioural data, we're also missing quite a lot of behavioural data. As you can see, if we pick this fish out here, it's been detected right at the far northeast corner of the array and then has come back southeast. So somehow that fish has got around the array or potentially through the array to be detected on this point here and to come back south. Similarly, this fish here was detected right in the middle of the array first and then was heading northeast. And again, we have another one here where the fish was detected right on the northern part of the array first and then detected heading southeast. So we're clearly missing quite a lot of information there, which is useful in its own sense because we now are able to understand that there is a lot more movement going on in this area than we thought, that there is a lot more circular movements or milling behaviour going on that these fish are doing. And if we pick out a specific fish to exemplify that, we can see here that the fish was first detected on the south part of the array, detected moving north all the way through to this point, before heading southeast and being detected on the southern part of the array again. But you can see here in the middle there's no orange dots. That fish has made it from the northern part to the southern part without being detected in the middle. And that's the same when it's eventually headed back north again. That fish has not been detected in the middle on its way back north. And although these are straight lines depicted here, clearly that fish is not travelling in a straight line. There is some sort of movement going on here that we are unable to see. But what we can see is that this is not a directed migration in this area. We know that the currents do not go in circular movements in this area, and that this fish is potentially doing something different to a directed migration. And then finally, looking at the inter-array detection. So this is looking at the detection movements between the Laxford and the Loch Torridon fish. What we have here is the percentage of movements in each direction of those fish. Just remember that the Loch Torridon fish, there's much fewer of these than there are of the Laxford fish. But interestingly, we can see that the Loch Torridon fish are generally going in most of the movements are in a north or a northeasterly direction, as we'd probably expect in a directed migration, whereas for the Laxford, the majority of movements are in the easterly direction and the northeasterly direction, with quite a few also in the southwest westerly direction here, which we don't see in the Loch Torridon fish, and that might be just due to sample size. But what you can see and also take away from this, that there are movements in all directions pretty much across the array itself, that it's not a consistent direction in simply a north or a northeasterly direction, that these fish are going southeast, southwest, west and northwest as well, that the fish are moving in all different directions. It's not an active or a simple straight line direction that we might have considered before. Finally, moving on to some temperature and depth data, and there's a lot more information on this from the 2023 presentation that is available on YouTube, but as we can see, these are the detections of these individual fish and the depth that they were. We had center tags on these fish, so the depth that these fish were at in the sea. You can see they're right in the surface waters, so all of them detected below within the top 1 meter of the surface, and we also have some temperature detections here. We can see this is about temperature of the water, so none of these fish have been predated by mammals that we can tell. So what we are seeing is natural behaviour or behaviour of real smolts. There's no evidence to suggest that what the behaviour is we're seeing is a tag that is inside a predator or a tag that is not actually within a smolt itself. And finishing off, what we have for 2025 is a grid of 50 receivers, which is currently deployed, and we've managed to extend the grid array in its original position all the way across to the main land of Orkney, and this is part of the Bluefish project, which is run by Edinburgh Napier University, tagging porbeagle sharks, taupe and skate. But we also hope to detect smolts here in the spring from colleagues who will be tagging smolts in Loch Carron. Hopefully those smolts will be heading north again, but unfortunately there'll be less fish tagged for this array going forward. And finally, just to finish off, I think the take-home messages here is that this is some really novel data that we have, that the fish are not migrating in a straight or a clearly fast migration either, that there appears to be some fish that are spending longer periods of time in this part of the sea, and that they are not moving in straight lines, and they are not moving all in one direction. There is a lot of circular movement going on, or some different behaviours going on than directed migration. What that is, we don't really have the data to say. However, the likelihood is that this is probably some sort of feeding that is going on as the fish are slowly making their way north. But again, this is some really novel data that we've been able to glean from these collaborations, and to be able to get this work done has been fantastic and has given us a real insight into what might be happening further offshore for these species as they're heading north. Thank you.
Hello, my name's Adam Piper from the Institute of Zoology, and I'm going to talk to you about some studies we've been doing for the last 2 years to look at the behaviour and migration routes of salmonids around wind energy development sites in the North Moray Firth. So the overall study aim is to provide an evidence base to assess likely impacts of offshore wind development on salmon and sea trout to aid future planning. We had three key focus areas. We wanted to understand, do juvenile salmon post-smolt swim directly north, which would be the shortest route to their presumed feeding grounds? If not, if they head east or south, we wanted to understand the amount of overlap between those trajectories and the wind turbine zones. And thirdly, we wanted to understand detailed behaviour of post-smolts within turbine zone areas and also detailed behaviour of sea trout. An overview of the study system. Each of these black dots is an acoustic receiver which detects the tagged fish as they swim by. We have this northern and southwestern Marine Directorate salmonid lines of receivers. In addition, the inset there shows Wick Bay, which is ZSL receivers, and through the river. I wanted to draw particular attention to the prepared project array in the wind turbine area, and really to highlight that how these large-scale, long-duration studies such as prepared allow things like our salmonid studies the opportunity to collect much more data than would be possible as a standalone project. We were tagging salmon smolts in the River Wick, in addition adult sea trout from the River Wester in a translocation study which I'll talk more about. In 2023, all of the receivers were retrieved, so we have the data from all of those, whereas in 2024 it's only these green dot receivers that have been retrieved, so I'll be mostly talking about 2023 data today. We're capturing salmon smolts as they migrate down the River Wick at night in April and May. We tagged approximately 100 fish each year with V6 or V7 ID tags. In 2024, 30 of those tags also had temperature and pressure sensors on them, which just allows us to see a bit more detail about the fish behaviour. Juvenile salmonids often experience high mortality rates in the freshwater component of their emigration. However, we experienced quite high survival. In 2023, 88% of smolts, and in 2024, 89% of tagged smolts survived to the first receiver line down in Wick Bay here, some 11.1 kilometres from their capture, tagging and release site. Reaching the marine habitat of Wick Bay, smolts passed through and out into the Moray Firth quite quickly. The histogram here shows you 95% of the smolts spent 24 hours or less in Wick Bay, and you'll see actually the majority passed through within the first hour or two from reaching that area. And the clock plots are to just show that the majority of this migration occurs under darkness. Most smolts that left Wick Bay, 36 were detected in the wider marine array. In the bottom left-hand corner there, it's the total duration of residency events in the bubble plot. They spent relatively small amount of time within the array, between 15 seconds and 3.9 hours. You'll see in the top right, this is an animation of the individual post-smolt tracks, and while some individuals passed directly through the array, there were also individuals that showed more complex behaviours, moving south and north again, or east to west. On to our sea trout studies. We would ideally be capturing adult sea trout out in the open ocean around the turbines. That's not feasible for a number of reasons, but we just wouldn't get our sample size high enough to be able to do the study. So we have to do the next best thing and capture fish in nearby marine habitats where we know the fish are aggregating. This is the bottom of the nearby River Wester, which I showed in that first plot, where we're doing sweep netting in June and July, capturing adult sea trout, tagging them for a translocation study. In 2023, we did a pilot study with 17 fish, and because that went well, we did it with another 49 fish in 2024. After the fish are captured, tagged and recovered, they're being transferred into these large tanks of seawater, food water from their capture site. The tags that are being used have temperature and pressure sensors on them, so we're able to get as much information about the fish behaviour as possible. The fish are being taken out to the turbine array and released into the water. We're doing everything possible to minimise the disruption stress to individuals. So even though they're being captured in salt water, we're also acclimating them slowly on the boat trip out there by pumping in additional water, changing the water within the tank from the surrounding water before they're lowered gently into the turbine array from the tank here and allowed to leave the tank volitionally. Sea trout were released in two groups, in the northern part of the Beatrice site here and in the southern part. And these plots are to show again the total duration of residency within the site, which range between 16 minutes and nearly 16 hours, but typically about 3 to 4 hours. This animation is individual sea trout tracks and really illustrates that they explored all parts of the turbine array. We know there's periods where individuals leave the site and come back in again, but quite complex behaviours seen here. This is an abacus plot of the raw data with time along the bottom and individual fish ID up the side to illustrate that fish were leaving the site and coming back into the site detected over an extended period. Indeed, time difference between the first and last detection ranged between 25 minutes and 17.25 days. We've got some interesting depth data from the sea trout tags. The majority of movement, 90% of depths, recorded in less than 12 metres of water, as we'd expect with sea trout, but we also get this classic sea trout diurnal pattern of them diving deeper in a bright, clear day and then coming up into the upper part of the water column at night. In summary, a substantial proportion, minimum 36% of post-smolts didn't head directly north, so these individuals have potential to interact with either current or proposed wind farms in the Murray Firth. We saw relatively short residency time within the turbine array of post-smolts, but we did see these complex behaviours for some individuals, so it wasn't just direct passage through. For the translocated sea trout, they appeared to explore or at least move around all areas of the turbine array. We observed those diurnal patterns. Really just wanted to emphasise that when considering potential impacts to the fish moving through these areas, we do need to be thinking about this complex three-dimensional behaviour within these areas. And for future work, the main focus is to get the outstanding receivers back in May 2025 to fill in these data gaps and to investigate the migration trajectories relative to the wind turbine features themselves, so those bases in the wind farms where the turbines are, and a hydrodynamic model for the site. I'd just like to thank all the people who have helped and supported the study, and please feel free to contact me with any further questions.